Y hay un último paper de este año 2026 que hicieron en China, unos chinos, muy buenos sobre el limfosoma bucal. Porque todo eso es
Okay. We are ready to start again. Well, are you already know Petar, our best friend? Now he's presenting his talk about Frankia. Well, this Frankia and other symbiotic microbes who work, spend their life together in the plant is a good example about what this kind of molecular conversation is being produced between one microbe and special olobionts, in this case plant. But this conceptually is the same model that we are trying to develop in the workshop. So Petar, you are welcome again.
Thank you very much. You are privileged we can discuss more relaxed on this way. If you have questions, just interrupt me and I can talk you more deeply about different part of my presentation. Feel free. I will try to speak very slowly with not so many complicated words that you can understand, and we can I can transfer this knowledge that on the subject that I'm working almost 20 years on Frankia symbiosis. So origin of symbiosis in Rosid is 100 million years ago started, and this plants, they lost this capability to be in symbiosis with bacteria for different reasons, because they reach a source of the nitrogen, and with time some plants, they lost the capability to live in symbiosis. However, the pioneer plants, they still keep this root nodule symbiosis with the two kinds of bacteria. One is gram-negative rhizobium that infect leguminous, and another is Frankia that can infect 8 family of plants. So Frankia is a specific Actinobacteria that living in symbiosis with plants, that they are very important in ecology of the environment, because they colonize this plant. They can colonize the land that is not so favorable to live, like some dunes, ashes. After glaciers are removed, they start to grow there. And they have very important economical and ecological role. In China, they planted millions of these actinorhizal plants just to protect extension of the Gobi to inside of the region where population live. So these plants are also important to treat some polluted soil. They have also importance for the for production of wood, and these are some examples that I presented just here, that you can understand importance of these plants. So this example that plants live with these bacteria, like alder, coriaria, datisca, elagnus, myrica, casaurina. So they don't look so well because they live in poor soil. I didn't show maybe alder. It could be something like 30 meters, very big. And all other plants, they are not so big, except maybe myrica, that is casaurina is here. that it can grow like 10 meters high. So now a few words about Frankia. So Frankia was discovered in the nodules of this plant, and we succeeded to cultivate outside of the nodules. And this bacteria is a gram-positive bacteria, belongs to the group of Actinomyces or Actinomycetota, and it is capable to fix nitrogen from air, and in pure culture, for example, Rhizobium is not able to fix nitrogen in pure culture. It must be associated with plant. This bacteria can fix nitrogen from air in pure culture. So we can grow in some media without nitrogen, and then in the culture we will find some hyphae. So these are cells specialized with nitrogenase inside. I will explain you later. And inside there is a nitrogen fixation. Nitrogen N2 from the air is transformed to ammonia. During the growth, when conditions became difficult to survive, this bacteria can form spore. They form specific sporangium, and inside there is spores that can survive very the conditions when bacteria has some limit to grow. And I will show you some other pictures showing some sporangium, like you can see here, large sporangium. How it looks under electronic microscope. You see that the spores are inside. And here we can see formation of sporangium, and at the same time, this is vesicle that is specific for nitrogen fixation. So, is it okay? So we can filament those cells. We have sporangium with spores, and we have specialized cells that have nitrogenase inside for nitrogen fixation. Okay. And now I will give you a little bit explanation about infection, how these bacteria succeed to infect plant, what's happened from the bacterial side and from the plant side. So if we look this is some roots, some lateral roots, and with a lot of small unicells, root hair, that just can have opinion about what's happened outside of the environment. So here we have a Frankia with hyphae, and then infection started that they must meet each other. So now model system is alder, Alnus glutinosa, and these are experiments made on this plant. So when plant grows, root hair can sense molecule that is secreted by Frankia. We don't know what is this molecule. We try to understand, but until today we don't know. We know a lot of things about these signaling molecules. We know that concentration, active concentration is 12, is a picomolar concentration. Picomolar concentration of these signaling molecules from the bacteria can be can provoke signaling on the plant. This is root hair that is already recognize this bacteria and starts to change. Thank you very much. So as a result of this infection, after 20 days, 21 days, we can see the formation of the nodule. Inside of the nodule, we have franchia that fix nitrogen. So, so here is summary. So if we cut this nodule here, for example, we cut like this. It is schematic presentation. So here we have soil with franchia is, for example, here. So this is root hair. It is one cell. And how they met each other? Because franchia is not motile. It cannot move. But root hair, it grows. They can touch. And there is some affinity of the root hair to the franchia. I will show you maybe here. Yeah, these are root hairs. You can see, and in the soil. And where is it? Yeah, I will. Okay, so when franchia is near to the, we add some extract of the bacteria, and we can see, we can observe that this root hair, it goes to the source of the signaling molecules, and then it makes retraction. You want to see one more? You see, it just goes, and then there is retraction. This happened during 12 hours maximum after we add signaling molecules. Of course, if we add bacteria, it will be the same. Okay, but we know that there is here there is no franchia, but there is signaling molecules from franchia, and we can dilute to 10⁻12 and still working. Very, very low concentration. Okay, I will go here back. So how they touch each other, I will give you a talk later. There is specific lectin genes. Lectin genes is special proteins that they can bind specific sugars. So plant has some lectins, but we found, I will talk you later, some lectins from the bacterial side. And when they touch each other, then there is the formation of the root hair, and bacteria goes inside. I will show you later such photos showing this. And what's happened from the plant side? You see here root hairs that they deform and so on. But immediately after adding these signaling molecules, we observed that there is a signal from the root hair inside of the cytoplasm and inside to the nucleus with calcium spiking. There is a receptor that recognize these signaling molecules. There is autophosphorylation and transfer of the signal inside of the cell. And there is a calcium calmodulin specific protein that activate transcription of the genes in plant that will allow bacteria to develop this nodule symbiotic organ. So we can measure pulsation of the calcium spiking. This is a transfer of the signal after we add the signaling factor from Francia. We know that it is less than 10 kilodaltons. We know that it is 1.2 kilodaltons, the size. It is thermoresistant. There is no UV absorption. We think it must be some kind of the sugar, maybe short lipid on it, but we don't know what is this molecule. This is an enigma for the moment. We know today that there is node factor in Rhizobium, but this is not, here we have no node factor like in Rhizobium. Mic factor that is important for microrhiz is not present here. So there is third signaling molecules in actinorhizal plant that are recognized by plants, but this is challenge to be discovered. What is this? Since we now have better chemical analytical methods, probably one day we will know what is this. For the moment, this is big enigma. So we just made some fractioning, and we still have this activity of calcium spiking inside of these cells. And we can see also deformation, but this is just shortly we just add this signaling molecule on root hair of alder. Okay, we skip this. So after 7 days, we can see something like this. All this root hair, they are deformed. They try to find. Everybody likes Francia, and every root hair likes to take it, but only a few of them they succeed to meet together and to make infection. So this is the root hair that is deformed, and there is specific place where Francia goes inside, and yeah, this is like Yeti. We call it like Yeti. Try to pick up the bacteria. So what's happened else from plant side? We succeeded to do a transcriptome of the plant. So we isolate total RNA from plants. In fact, they are infected by Francia, and then we sequence all this gene expressed during symbiosis, and we were succeeded to identify a common symbiotic pathway in alder and also in another plant. And here are genes that we identified that they are present during infection and also in symbiosis with Francia. So we know also now that there is Francia receptor for to receive these signaling molecules. The transfer of the information by different genes that will activate transcriptional factors that will activate expression of the genes. So what's happened from the bacterial side? First of all, we sequenced many genes, many strains of Francia, but our model is Francia aldi that you see the results before. We have other Francia strains. Today we have something like 50 genome sequence because it is very easy to do this. What we can see that some Francia, they have very reduced the genome size, and this Francia we are not able to cultivate in the laboratory. We don't know why they miss some genes, and our conditions to cultivation, they are not good. But other Francia we can successfully cultivate with the very poor carbon source like propionate, so C3 carbon source, and with other salt and nothing else. No nitrogen. It will form vesicles and it will fix nitrogen. So this was starting point for our analysis, and we also do this transcriptome analysis in under different conditions when Frankia is in the nitrogen-rich media comparing to the nitrogen-poor media, or in symbiosis. And then we can compare conditions, what is important for each growth conditions. So here are results of expression of the genes in Frankia when they grow with nitrogen, without nitrogen, and in symbiosis or not. So we found different gene level of expression of different genes, and we found some specific genes related to nitrogen fixation. Yesterday I spoke that some essential genes, they are always located or near to the origin of replication. Here is origin of replication, and we found that nitrogen fixation genes, NIF cluster we call it, they are almost at the beginning of the replication. So this cluster is essential for bacteria, and it is preserved and it is kept near to the origin of replication. We have other cluster genes, SHC cluster, HOOP cluster, and the second cluster of SHC. So these are genes important also for nitrogen fixation, because these clusters, two clusters, SHC1 and 2, they synthesize squalene-Hopan structure that are important for protection of the nitrogenase from oxygen. So this cluster synthesize these layers of hopanoid. They protect nitrogenase from oxygen. There is also sulfur cluster. This is important to form metalloenzymes, because SAF cluster is important to import sulfur inside of the cell and to make complex with the iron or molybdenum, and to be incorporated in not only in nitrogenase but also in another iron sulfur metalloenzymes. So is it okay? You can follow? Yeah. Who didn't got chocolate yet? So you can share. Okay, what I'm with time, I'm okay or not? Yeah. Okay, so I spoke with the lectins genes in plant, and here we found two lectin genes that they are present in the Frankia genome. And the question was, are they important for infection? And the answer is yes, but we must confirm this. So these two lectin genes, they are highly expressed during infection, and we try to clone these genes and purify, and also to make some reporter genes fusion with this gene, and to see how it will, where it will be during infection, and how we will succeed to have nodulation. So here is just short explication that we succeeded to make fusion of this gene and express in E. coli. So we made fusion with the reporter gene. So this is glucuronidase. So is enzyme that is present in bacteria, but not in plant. And it can degrade some specific substrate that can give some coloration, and we can see where it is a protein. So we also confirmed that this gene is, these two genes, that they are also expressed during the in nodule, also in pure culture, and we confirmed that they are expressed in all these conditions. So we started to go with experiment. These are three-dimensional structure predicted from this protein. So what we found that it looks like crystalline protein, and this protein is similar to ricin B protein from plants. Ricin, you know, ricin B and ricin A. Ricin A is very toxic protein. Ricin B is not toxic protein. It just bring ricin A, that is very toxic ribonuclease that we can be killed by this toxin, ricin A. But ricin B is similar to this model, to these two proteins, and also with the toxin protein from Bacillus thuringiensis, and it is capable to, so this is another projection of the protein. It is stabilized with 1, 2, 3 SS bridges, and it can bind to the surface of the root hair. So this is root hair. You see these cells from the before. We mix frankia together with extra purified protein. That is, it has this glucuronidase activity, and we try to make infection. So what's happened? We can see here that this protein, what is blue is inside, but inside is frankia going to the root hair. Maybe you cannot imagine. You can see that there is hyphae is going inside of the root hair, and also we mix this protein with the pure culture, and we found that this protein keep these cells also together. This is a role also to keep mycelium together. If we apply only gas protein, we cannot see coloration, but this is very intensive coloration. So we think that this is some molecular glue that keeps cells together and also can attach to the root hair and to help bacteria to infect root hair. So we made some experiments adding this extra protein to frankia culture, and then we can increase the number of the nodules. So we help frankia to succeed to attach to the root hair. And finally, we can obtain some nodules. So this is a picture of nodules that we successfully infected with the frankia, and how it is after 21 days. You see nodules here and root hair that they are a really nice picture. We succeeded to see with the formation of the root hair here. Are you okay? Okay. So, so now a few words about vesicle and nitrogen fixation. So these are specialized cells with the nitrogenase inside, with other protein that I will give you talk, and also they must be isolated from oxygen. Why oxygen? Because oxygen is very toxic for iron, it will bind to the iron, and then activity of metal enzyme is stopped. So this nitrogenase must be protected. So, as I told you before, I didn't spend time to explain you the results of different gene expression before, because it's too complex, many genes overexpressed and so on. I just made a summary here. When we have a pure culture without nitrogen, it means BAP minus, in comparing with BAP plus. And symbiosis, this means in nodule with plant, we can compare the expression of different genes that they are part of the nitrogenase structure. So we can see when we have no nitrogen in the culture, all these nitrogenase particles, genes, are highly expressed. They are very, very expressed, highly expressed in nodule, more than in pure culture. Okay, you see the 46, 49 for some proteins comparing in pure culture. So in nodules, this is very, very, very active machinery, and we can see this comparing this transcriptome in different conditions. So what is nitrogenase? So this is a metal enzyme complex. It contains molybdenum and iron as a essential active site. So this is tetramer, two different dimers. There is alpha 2, beta 2 heterodimer. Alpha heterodimer is coded by NIF-D gene, and beta by NIF-K. So this is active site with the molybdenum, iron, and sulfur center. And there is also iron sulfur complex in the structure. So, and this enzyme can fix nitrogen and from the air and transform in ammonia without high pressure and high temperature, at room temperature. Okay, so this is just a short presentation that there is a ferredoxin that participate in the electron transfer, and we have finally production of the ammonia and hydrogen. So hydrogen is very rich in energy. So what we can do with this hydrogen, bacteria just remove this hydrogen and use it to for the energy production. And to do this, it has hydrogenase uptake system that just keep this hydrogen, and there is two clusters that one is expressed in nodules in plants, and another is expressed in the pure culture. And I told you before, SHC clusters, they are involved in biosynthesis of hopanoids. So these are molecules that form these layers around the vesicle, and they protect this complex from oxygen. Oxygen cannot pass all these layers. So if we look this structure with the, this is a freeze etching pictures. When we scratch surface of this structure, we can see layers of the hopanoids here. Okay, so other things that we try to see what's happened on the plant side, on the bacterial side, how bacteria can just pass this barrier, because the root hair is protected. There is some cellulose outside and so on. And we found some cellulose degrading enzymes. I will just show you here. So this is only plant, small plant on the agar. This is coloration with Congo red. So this is Frankia. You cannot see it's on plate. And here, if you look, we apply the Frankia on this root, and you can see the decrease of the some hollow around here. Maybe it's not good. And so this is cellulase activity of the Frankia. So for a very short time, there is activity of the operon that called cellulase operon, and it stopped to work very shortly. So we think that during infection, there is one moment that this cellulase is very important just to make hole, go inside and stop. And plant can repair this damage, and bacteria goes inside of the root. Yeah.
When you have the nature, there are a lot more bacteria around the root.
Yeah.
What could be done if Frankia will be cellulose to make the holes? Okay?
Yeah.
Why not another bacteria could go inside the plant too?
This is a good point. We identified also other bacteria. We know that there is cheaters, and also some bacteria can go together if this culture is contaminated, like Micromonospora can go inside. We made experiments and we found Micromonospora, another Actinobacteria. We don't know what it's doing inside, but they are present also. We can isolate from nodules and yeah. But what is interesting, that plant is quite selective. We have many different bacteria in soil, but only few species can go inside of the root. Okay. Okay, now I show again this slide. This is common symbiotic pathway. And now question that you asked me before, can we have all plants infected by actinorhizal plants? So this question was, we asked ourselves also, okay, why other plants are not symbiotic with the Frankia? For example, Betula pendula or other plants, only just 8 group of plants and not other. So to answer this question, there was very big project. That summary is shown here. So we sequence all these blue marked plants, and we compared the genome of all these plants. So we try to find all these genes that they are important for signaling and for nodule development. And if you can see here, plants that they are not able to develop symbiosis with actinorhizal plants, they miss some of the genes, some of 25 genes that they are important for symbiotic development. So most of them, they have no NIN, so this RPG, and some of them, they have no receptor. So you can see here is answer on the question why some plants are not able to live in symbiosis with Actinobacteria frankia. So this is the article is one of the most cited in our field, is published in Science 2 or 3 years ago. So this is the answer or your question. Okay, so now when Frankia is inside of the plant, what's happened? So we found from the transcriptional analysis of the plant that plant produce some strange kind of transcripts that they code for very short proteins, 80, 60 amino acids. It's not big protein, but we found something like 400 such genes. 400 is too very, very, very much. And one of them, we call it AG5. It was very, very present. Transcripts was very present. Then we decided to produce this protein and to produce antibodies. And when we applied antibodies against AG5, we found all these greens fluorescence belongs to presence of this protein AG5 in nodule. So what is his role? Yeah, so this is different position in nodule, and you can see this is infected area and so on. And senescence. So this AG5 is associated with Frankia cells. So when we add this protein to the pure culture of the Frankia, we found that at some concentration, we can increase nitrogen fixation, but not with, if we increase more concentration, more than 3 micromolar, then Frankia died. So there is some optimal concentration of the protein that Frankia fix very well nitrogen. So this is this 0.3 concentration of the AG5 protein. So this is a protein rich in cysteine-rich protein, and then we measured all parameters around. And what we found, that contrary to our idea, the concentration of the carbon dioxide in the culture dropped down. And my postdoc, Lorena Caro Garcia, at this time, she's first author of this article, she told me, Petar, I'm sorry, I made some mistake. Let's do again. I said, why? She said, you see that the CO2 is dropping down. I think there is some that I didn't close well system, and I tried to repeat. And she made the second time, and she said, Petar, I don't know, I'm crazy, but it's the same. I made the same mistake. I said, no, you didn't make a mistake. This is very interesting thing. And she said, no, I will do this. And she made repeat in 2 replica with 5 tubes and so on. And also she got the same thing. Because we know that, we think that the bacteria is saprophytes, they will produce CO2. But in this case, I say, no, this is very interesting thing. So we try to explain what's happened with CO2. So there is 2 explanations that I will show you later. I still have a few times. Okay, so we repeat this experiment in pure culture without AG5, and we see decrease of the CO2, and here also during the nitrogen fixation. And then we check the literature. We found that some people, they try isolated Frankia, they add CO2 and it can stimulate the growth. So this is correlated with our results. Then I try to understand what is what's happened, what is important. Some other experiments, experiments, they show that other nodules fix CO2, but it could be also related to phosphoenol pyruvate carboxygenase activity of the plant. But we try to dissociate this thing, and we try to see phyloprofile of the gene expression in Frankia. And what we found two genes that code for carbonic anhydrase. So this is first enzyme that took carbon dioxide and put it in the water and just dissociate. So we started to be interested in two genes. So shortly, what's happened when nitrogen is from the air is fixed, it produces ammonium and OH. So this OH just increase pH. So we are under basic conditions, but for optimal activity, we need a pH about 7. So this carboanhydrase just introduce CO2 just to neutralize the basic pH of the nitrogen fixation. So we can see this also with the specific coloration in nodule that it's happened. I will show you later. But also there is a new thing from metabolic analysis. We found that this pathway of metabolism of the propanoic acid, because Frankia grow on C3 carbon source, we found in the genome all these genes that they allow to propanoic acid goes to the succinyl coenzyme A and go in the Krebs cycle. There is two important things. So there is biotin that is important for fixation, for adding CO2 to the to produce D-methylmalonyl coenzyme A. Then epimerase is go to L-methylmalonyl coenzyme A, and specific mutase just transform to succinyl coenzyme A, that is substrate for the Krebs cycle. Two vitamins, biotin and coenzyme B12 is very important. So coenzyme B12 is a cyanocobalamin, and so we know that this is a very important vitamin, and we also found that cluster for vitamin B12 is also present and near to all these genes on the genome. Yes.
This vitamin pure culture.
Yes.
Without plant, I think.
This is without plant. Yeah, this is in the pure culture with only propanoic acid as only carbon source. Okay, C3. So just remind you that our mitochondria has operating Krebs cycle, and one of the essential pathway is this pathway to degradation of C3 carbon molecules. If we have no these enzymes, we have some metabolic disease, very, very difficult metabolic disease, accumulation of the C3 molecules in the organism.
So, one more. The energetic ratio between amount of carbon source and ATP.
Yes.
Is obtained from the 6th cycle, no?
Yes, but after the energy is coming from the degradation of the C4 unit, because this bond you have three carbon. You have a release of the energy from these 3 carbon bonds. yeah, there is, it energetically is not rich, but this is way how bacteria survive, because there is no other carbon sources. Yeah. Okay, and the second part that we found that a lot of, by metabolic analysis, there is a ornithine and citrulline in the metabolism of the franchia, and we found also that this gene is very highly expressed, carbamoyl phosphate synthase, synthesizing carbamoyl phosphate. And just to synthesize this, the bacteria need one molecule hydrogen carbonate. So also this is the second part where carbon dioxide is incorporated also in organic molecule. So this is just show you the pictures when the nodules are they keep the pH with some specific coloration, low pH in vesicle, and so this is another one. And in the genome of the franchia, we found a third one, carboanhydrase gene. One, it is related to some membrane protein, and these two that we cloned and we sequenced and we expressed. So this is metalloenzyme that contains metal incorporated between cysteines and asparagine and histidine in the structure. We show that all these genes are expressed. Some of them, they are specific for nodules, for FRAL1, 2222. Another one is constitutive. It just is housekeeping gene, but also carbamoyl phosphate synthetase. They are expressed specifically for the in the nitrogen pore media, and some specific for the nodule. So these are two genes structure that we made cloning in E. coli, and here is a native gel after expression. We add some specific substrate, and as we add CO2 in 1 or 2 seconds, the pH change, and we can see a yellow coloration. So this is a purification of these two carboanhydrase. So here and here. So this is a expression lysate of the bacteria. We purify these two proteins, and we made some biochemical analysis, determined the kinetics of this molecule in comparison with the human carbohydrates as a standard. So we know some specificity of these proteins, and oh, sorry. Okay, and so this is for the end. This is last results, is not published yet. I finished this just before going to Concepción, and I just like to share with you. This is not IE. This is my work that I made the synthesis of the proteome in nodule. So here is a bacteria living in cell of the plant, and here on the left, on the red, there are proteins that we cannot find in symbiosis in plant. When there is no bacteria, all these red proteins, they are present, but here they are not present. They are suppressed, they are less abundant. So what we can see here, this is Krebs cycle of plant, and this is Krebs cycle of bacteria. When we have infection with bacteria, plant became lazy. It stopped to use her Krebs cycle. It produced just antimicrobial peptide that I showed you before. They make hole in the membrane of the poor plant bacteria, and they leave, and bacteria works for them. Yeah. So not only carbon cycle, but also nitrogen fixation, and also CO2 fixation. So poor bacteria is working for the plant. So with this, I will finish my talk, and some take-home message that Frankia is not only nitrogen-fixing Actinobacteria, but also can operate as a CO2-fixing bacteria. And also that is an important factor in CO2 and nitrogen cycles on the Earth.
One thing, this is amazing, really. The CO2 fixing, of course, is not the visco.
No, I told you that there is a, this is a, yeah, here you have fixation with the, to synthesize carbamoyl phosphate synthase. This is one point, and another point is here, propionyl coenzyme A carboxylase to do dimethylmalonyl coenzyme A. Yeah, here, this CO3 is here, and in succinyl coenzyme A, and in information of carbamoyl phosphate.
As I understand, the bacterial CO2 fixation is the opposite. It to work like a buffer system.
First, there is a buffer system to keep the optimal pH, because ammonia is protonated and give just basic conditions, because OH is increased, and also just protect, because this yeah, maybe in another one, because a nitrogen ammonia is a gas. If it is not protonated, it will go outside. It passes membranes, and bacteria must keep it. And when it protonates, it just keep like in solution. Okay. Okay. So if you have questions.
Open. Any questions? Of course. Go ahead.
Thank you so much again for this presentation. As a biochemist, it's always exciting to see some metabolytic reactions. So it was like, wow, there's a lot of work about these bacteria that, yeah, are exciting. My question is about the symbiotic interaction between the plant and the Frankia. Is there any specific event that determines like the efficient interaction between these plants? Like it's determined to the like why the plant interact
with this bacteria?
Yeah, and not other. Like
I don't know if it was from the beginning, but I started with the signaling molecule. Not all bacteria has this signaling molecule, and the plant will not react. If you add the E. coli, Pseudomonas, or some other Streptomyces, No root hair deformation, only frankia.
Yeah, I see. Yeah,
Only
sorry for
frankia.
that.
Yeah.
Thank you so much.
Any question?
Thank you for the presentation and for us responding my previous answer. But I have two more.
Okay.
And one of them is, if this is a bacteria that can be free living in the soil, right?
Yes.
Is it possible to just put this bacteria in the soil and the rate of nitrogen fixation, would it be better? Like we can improve the nitrogen in the soil by just putting this bacteria there?
Yeah. We do this experiment, and the problem is that bacteria need some carbon source.
Okay.
If you put in some poor soil without source of the carbon, nothing will happen. So it must be some carbon source inside.
Okay. And sorry.
Yeah.
And the other question was about the why some plants cannot do this symbiosis. And yeah, and there's a table where the genes are like marked that some plants doesn't have
Yes.
it. And with transgene, we can put those genes in the plants and technically
Yes,
would work,
yes,
right?
yes. There is big project that what is the name of the Microsoft? Bill Gates give big money to one international project to introduce missing genes in some plants that they have economical importance,
Yeah.
to restore symbiotic pathway, and to have these plants with the symbiotic bacteria, just to avoid to add some source of the nitrogen. And this project is still on. The people are working on. They try to introduce to the rice and also the to other important plant that they have economical value.
Perfect. Thank you.
Yeah.
More question? I have a lot of questions.
Yeah.
So, microbiology behind what's the model of the gene expression control inside the plant and inside the bacteria? For example, when you talk to us, many genes are expressed and others are closed. So, my question is, what kind of mechanism of regulation of transcription is involved on quorum sensing or sigma protein?
Yeah, there is a two-component system. There is a serine threonine kinase, and you have a receptor that can receive the molecules, and there is autophosphorylation and signal transduction to the response regulator that phosphorylate calmodulin, calcium.
I think it's the same quorum sensing gram-positive model for.
This is not quorum sensing, so this is the signal transduction by a two-component system. Yeah.
Now, but the induction, the signal is coming from
Bacteria.
Okay.
Yeah, and what we know that it seems that this signaling molecule is not is present all time in the culture of bacteria. We try to increase the production, but we didn't succeed to see. We made the serial dilution of the culture of bacteria, comparing with some plant extract adding to the culture, and we have serial dilution, and until 12 to minus 12 we have activity, but not in the same experiments with plant extract and without plant extract. So we conclude that this molecules is somehow constitutive, maybe some part of the membrane. We don't know what is this.
So finally, if you compare it with the Rhizonia model, which is the main difference? Rhizonia, they will,
Rhizobia, there is they use a common signaling pathway. The receptor is different. Is not same for node factor, and definitely is different. Yeah, is not factor present. We try to do this with node factor, but not actinorhizal plant don't recognize node factor. It is different, not a MIC factor.
okay.
So it is specific. So person who will identify this molecule will be very famous.
So, but I look to the oxygen as needed for this aerobic metabolism.
Yeah.
But in 11, so we use left hemoglobin oxygen transport.
Yeah, and this some actinobacteria like CCA3. Is this? Okay, here. So this bacteria CCA3, it has no capacity to produce this protection for the nitrogenase, and in this case, plant has plant hemoglobin to capture the oxygen. Yeah.
Fantastic. Any more questions? Of course. Oh,
Thanks, Professor. Thank you, Professor, for your presentation. I have a question. I will try to explain that. If you put the last diapo, and oh.
This one?
No, the other.
Before? No.
That's the conclusion. Okay. I kept my attention the last sentence. Look, in eukaryotic cells, there is a moment when the cell don't go to the Krebs cycle and stop in the glycolysis.
Yeah.
And that metabolism is called the Warburg metabolism. And maybe it's possible the plant cells is eukaryotic cells cannot go to the Krebs cycles and use this bacteria for his mitochondria.
Yeah, what we saw from these experiments, we know that in cell there is a Krebs cycle of the bacteria operating, but what we know, I didn't present here we know that these antimicrobial peptides, they make a hole in the membrane of the bacteria, and there is metabolites from bacteria going outside. We published this already. I have no time to discuss about this. We know that there is a efflux of this metabolome. What we know that there is a C4. So this is decarboxylate transporter is highly expressed in bacteria, and we think that there is some molecule like C4 carbon is imported in bacteria, and it uses this as a carbon source, but produce amino acids rich in ammonium like glutamine, glutamate, and so on. That is important as a source of the nitrogen, but also there is a transporter of the nitrogen, ammonia, that is present in a plant that probably share this amount of free ammonia ions inside of the cell.
Thank you, Professor.
You are welcome.
More questions? I have one. Last one. I mean, I'm thinking Francia when we.
Sorry.
Are you? Of course.
Hi, Professor. Thanks for your presentation. I had a question because I got lost at the carbon dioxide graphic because you were talking about the lower concentration of the carbon dioxide, but I didn't understand where you measure that lower concentration, if in the roots
okay.
or in a medium or soil.
Okay. Imagine we are bacteria, we breathe, so we consume energy and our body produces CO2. It is released by expiration. So it's the same with bacteria. We have for this experiment, we have in the flask bacteria, in the liquid media, and the upper part is air. Air, pure air with the nitrogen, oxygen, and CO2. We start to measure amount of the carbon dioxide from the beginning and until end, and everything is closed. Nothing can go outside. We just took some needle to take 100 microliters and just to go to the detector to see the amount of CO2. So this is air that is in the culture, but all system is closed, so there is no CO2 from outside. So
Yes,
this is bottle, very well closed, and with possibility that we can take just a few microliters for GC analysis or I don't know which other gas chromatography analysis. Yeah.
Okay.
Yeah.
Thank you.
Yeah.
Thinking in the life of Francia in the field, I think outside of the plant. How do you can imagine this life in terms of Francia is surrounded by a lot of different kind of microbes. Maybe some kind of collaboration between Francia and other ones to think, and then bacteria can provide some specific carbon source.
Obviously, what we know from the genome structure is that when we look E. coli, we grow E. coli, we had, I don't know, a rich media, we had glucose, and everybody's happy, it grows very well. But Francia didn't grow on glucose. We cannot grow on glucose. We think we can grow on some citrate, but Francia didn't grow on citrate. So it is very specific. So it will grow on propionate, on the fatty acid, but not on the glucose. There is no transporter for the glucose.
Oh, there is not a glycolysis.
And there is a pathway for gluconeogenesis, but not for glycolysis. Inverse pathway,
special,
yes. So there is a gluconeogenesis pathway that we identified, and there is production of the pentose phosphate. There is no Ettner-Dudorov pathway, but for pentose phosphate there is. It must be because it can grow only with propionate. You add propionate, nothing else. Of course, you need to add minerals, potassium, sodium, calcium, and microelements, and this will be enough to grow. So
just one thing. When you think on Francia living inside,
Yes.
We have learned that produce like nitrogen fixation.
Yes.
To CO2 consumed.
Yes.
There is evidence or experimental evidence that could be protect against pathogen, for example, as biocontrol too.
Yes, this is good question because I didn't speak about secondary metabolites. And what we found that alder, the plant, it has no parasites that can eat leaves. And when we found it's too many data, it's difficult to give you answer. I don't know, I have no here. Doesn't matter, I will try to explain. When we do metabolomic research, we identified molecules present in the culture and in the bacteria, and what we found GABA, gamma aminobutyric acid. This is an inhibitor, important inhibitor, and we think that this production of GABA can interact with the nerve system of the insects, and because of this, we think that there is no insect on this plant. One of the reason, probably there is also other, but this is one of the explanation. And of course, we found some production of the geosmin, and we know also that the geosmin has specific effect on the brain of the insects. Insects don't like geosmin. There is an effect on the if you give Drosophila two sources of the food, one with geosmin and another without geosmin, and Drosophila will not go to eat this food that is rich in geosmin. It will go to another. So we think that Actinobacteria conserved this evolutionary biosynthesis of the geosmin just to protect them for the carbon source.
Okay.
So not other is telling all other species, please don't come here. This is our territory, and the insect will not like to go to this place to look for the food. Contrary, where yeasts are present, then they will go to this place. Yeah. Okay.
Perfect.
Okay. You took. Everybody took the chocolate. Yeah. Maybe we. If you have more questions, we do some small break.
The second one. Let's take 10 minutes.
Yeah.
Walk. And then go.
Yeah. Okay. Hola. Hola. How are you?
How are you?
Fine. Yeah.
We have some special better food than yesterday. Which one would you like to eat?
Do you understand? This is soup with meat,
Okay.
potato,
Okay.
some vegetables. This is chicken
Yes.
with potato.
Mm.
This is also potato. But this is chicken. This is pig.
What is pig?
Meat.
Fish.
No.
A pig. Ah, okay. And this?
This is bowl. It could be vegetables with chicken
Okay.
or eggs.
Oh, this or this is fine.
Yeah.
Yeah.
The soup or soup?
This is what is soup of
Soup
with.
with meat
Ah,
in
okay.
little.
I can take chicken.
Chicken?
It will be much easier. Yeah.
Belén. Ah, ya. ¿Te lo escribo en el WhatsApp?
Yeah.
¿Él no?
Entonces, hay mucho que conversar.
Hola,
Sé que-
me
¿cómo te
llegaron
fue?
cosas de marca ayer.
Ah, ya.
Así que ahí
Pero la
cheque.
factura todavía no te llegó.
La factura me llegó física. La
Ah,
guardé.
ya.
Pero,
Pero pide la electrónica.
voy a pedir la electrónica, le voy a pedir la electrónica y le voy a pedir que me manden los datos, todo eso. Le voy a pedir la electrónica también. Pero me llegó física, así que la guardamos ayer. Las cosas, algunas cosas con Carolina las guardamos ayer.
Ya.
En la mañana. Así que eso. Así que no.
This is for you.
¿Ese quién? ¡Ay, eres tan linda!
Para que sea vegetariano, podría ser un bowl, pero con huevo, en vez de pollo.
Este es cuando fuimos a comer ayer.
No, si es para mandar a pedir para que no se haga.
Ah, bien. Sí,
¿Ah?
Se tiende a juntar todo el equipo.
Madagascar.
Hola,
¿Cómo estás?
muy bien,
Excelente charla.
Sí,
gracias.
compleja.
Sí.
Complejo entender el mecanismo.
Interesting. They shows me data
Ecuador.
and Drosophila.
With the jasmine.
With the jasmine, yeah.
¿Hay café afuera?
Hmm?
If you want coffee.
Ah, okay. I took one. This is okay.
I need one. ¿Tú quieres tomar coffee?
Me tomé un café en la mañana, me pedí un expreso y me comí
A niños de distintas localidades comunas para ver si había algún patrón
Pero de qué se va eso
Sí,
pudiera estar generando algún tipo de
súper rico. Así que aproveché. Estaba tarde como que lo busqué y me pedí el lugar rápido.
Yo estaba cerca del tema de
Hay un coffee break afuera.
Okay.
Are you working in both sides?
Yes.
Black and
Yeah. Can we start? We have just. I have how much? Yeah. Okay. Yeah. Okay. Thank you to be here. You tasted all chocolate. Okay, so my second presentation today will be Actinobacteria, that is known as a human pathogen and animal pathogen. And I will give you a short talk about this specific Actinobacteria. So history of this bacteria that is called Nocardia is goes in 1888 with that Edmond Nocard, the French veterinary, discovered this bacteria as a agent that provoke infection in bovine. And in honor of his discovery, they call it a Nocardia, this group of the bacteria. So yesterday I showed you this these slides. I just shortly showed it here that this Nocardia belongs to group of Actinobacteria that evolved 3,000 million years ago, and they are mostly soil bacteria, Nocardia. You see on the top. And some of them they are pathogenic to the humans, animals, and I will give talk about specific Nocardia that infect humans. So this will be not so nice to see. I apologize, but just to show you the disease that this bacteria can provoke with humans. So there is a skin infection. If you got injured somewhere with the presence of this Nocardia, you can have some skin lesions, and also if you breathe the Nocardia from the air, you can get some pulmonary infections, and also this pulmonary infection can disseminate in different organs, in bones, and also to the brain. And then we talk about infection of the CNRS system. So different kind of Nocardia are known today. Several of them, they are specific for the skin infections, like Nocardia farcinica. Others are more specific for lung infections and also dissemination to other organs. And as I told you, every organ can be infected, like liver, kidney, eye, and and bones. So here you can see different culture of different species of the Nocardia that we can isolate from such nasty infections. So here infection of the skin with the Nocardia farcinica, especially in the people that are working in the field when they got injured, and this disease can develop. So the Nocardia is filamentous bacteria, and you can see here coloration with the specific dye, and they can form specific also cocci during the growth that it's shown here. So it can change the forms. As I told you yesterday, if there is no rich media without some amino acid, it can be like unique cells. So it may change the form, and this probably is advantage of the bacteria that can be transmitted in the organ that in host when they infect some animals or humans. So different stages of pathogenesis are presented here. We can got nocardia if we inhalate this bacteria by air, maybe on PT 2.5 particle or PT 10. So we can also get, if we get injured by some spindle or so on, if we can introduce in the organisms. And then there is also the adhesion of this bacteria on the surface of the cells. Here I just show that a specific group of the family proteins, they are important for adhesion, that I will give some more information later. And then immunological system try to define our bodies against nocardia, but these specific molecules from the nocardia called court factor just protect the bacteria to be destroyed by macrophages. And also other system of resistance, SOS resistance, superoxide dismutase and catalase neutralize the effect of the immune system, and then they can survive a long time in organisms. So I'm going to talk about Nocardia syriacis georgica. These are colonies growing on some media, and you can see how they are nice, but they are nasty. This strain was isolated from human infection, from a person that died from a fatal systemic infection caused by this bacteria, and this bacteria is GUH2, Georgetown University Hospital 2, the patient number 2. I didn't put this information, but this is from human infection, and this is model organisms for a long time, and I was working on this for many years, even today. This is not my main research because there is group working in France in my lab, and when they need some special help, I'm always present, and I continue to follow to help them from bioinformatics part, from experimental parts, and this was my postdoctoral stage in California. I was lucky to work with the person that is important for the nocardia field, Blaine Beeman. Here, Professor Blaine Beeman from University of California, his wife Lovell, me and my wife. She was working also on nocardia. So. So when we made experiments with this bacteria, we used some mouse model, and on this graph we can see presence of the bacteria in the brain of the mouse. When we injected the bacteria in the tail, 2 hours after injection, we can find bacteria in the brain. How it goes there, we don't know. but we can isolate 2 hours bacteria. So on this graph here, we can isolate from the brain of the mouse bacteria. We can cultivate, they will grow. After 2 weeks, we cannot isolate any more bacteria. So mouse became free of the bacteria in the brain. No cultivated bacteria. So we can tell, okay, bacteria try to infect mouse, but mouse resist, his immunological system recovered from this infection. But it's fine. And there is some behavior of the mouse after this infection. So there is hemiparesis and head shake of the mouse. So it was specific related to this infection. So I will try to show you. Okay, this is a healthy mouse. It's very curious. It's just go slowly. Just try to, it's stressed also. And it just try to discover this space. But now we have the mouse that was infected with nocardia, GOH2, and we can see this movement, head shake, okay, and special tail position, and doesn't go straight. It is a very, very specific head shaking. So it is very disoriented and doesn't want to go to discover the place, and it's completely confused, and finally it will I think it fall down after few. It's go. We never saw. Oh, sorry. Mouse that is going back. Okay, so these are the group of the mouse that they show the similar phenotype. You see, once more, the specific head shaking, and then we try to apply L-DOPA and carbidopa. So this is the treatment that people that they have Parkinson's disease, they use this molecule, and we can stabilize the movement of this mouse with L-DOPA and carbidopa. So they have the same response like people with Parkinson's disease. Okay, so they are calm. They don't. They have no shaking like. But L-DOPA, we know it. It is degraded quite quickly, and after 2-3 hours, they start to have the same symptoms like before. So what we know about nocardia, that nocardia is filamentous bacteria, and it can adhere to the eukaryotic cells. And here different electron microscopy pictures made by Blaine Beeman that showing that that bacteria use tips to go inside of the eukaryotic cell in capillary on some culture cell. it's very important that it grows inside of the cell. So we know later we can explain some reason why this is like this. So this bacteria can induce apoptosis, cell death. And so this is the reason why these mouses, they start to have this phenotype, because we know that there is apoptosis of the specific cells in the brain infected with this bacteria. So we can have this effect also with some culture filtrate, because bacteria secrete some signaling molecules, some toxic molecules that can induce apoptosis and degradation of the specific PC12 cells that they have a dopaminergic receptor on the surface. And we can induce this by only by culture filtrate without bacteria. So in the experiments with the monkeys, we can compare appearance on specific Levy bodies in the human central nervous system, that people that they have Parkinson's disease, comparison with the monkey brain infected with the GOH2 strain. So we have specific Levy body-like structures. We don't call it Levy bodies. It's related only to the human Levy bodies. We call it Levy bodies like proteins. So this was an experiment that Blaine Beeman made with monkeys many years ago, and after he stopped to do this. What's happened? What we have idea about what's happened? We think that the system of degradation of the proteins is blocked somehow by a system of degradation of the proteins in the cell that is related with the ubiquitin and the three enzymes that mark cells, that mark proteins, and finally they should be destroyed in the cell. With something that is produced by nocardia, we block somehow this system of degradation, and this result of this inactivation in this formation of alpha-synuclein as a protein that is present in the cells. So this is definition of the Parkinson's disease in the human, but we have the same precipitation of the protein in experimental animals, and we think that bacteria produce some specific inhibitor type epoximycin, and I will I will show also here that some nocardia induce also cell death that is specific also for the brain. Here is model with the HeLa cells, because it was most easy to do this experiment on the HeLa cells. And when I turn back to France, I continue to follow the research on nocardia, because at this time in US was no genomics of nocardia, and I started to develop this in France, and I made a proposal to sequence the genome, And this was accepted, and now we have the genome sequence of this bacteria, GOH2, that is about 6.1 megabases. There is about 6,000 genes, and genome is circular, and there is about high GC contents. Number of coding sequence is about 5,500, and it's very, very dense with genes that these bacteria contain. So we try to understand what are virulence genes present in this organism, and here we found that genes coding for fibronectin binding protein is known as a fibronectin binding protein, is the same enzyme present in Mycobacterium tuberculosis, and also in another nocardia. It's specific, like AG85. This is a specific antigen in Mycobacterium tuberculosis. If you are positive on infection with Mycobacterium tuberculosis, you have antibodies specific to this specific antigen. So these are proteins involved in biosynthesis of the cord factor. Cord factor is a trehalose dimicolate. So this is a trehalose sugar with long chain of the fatty acid that are specific for the Mycobacterium and for nocardia. This long chain could be 60 carbons to 80 carbons long, and this is specific for this bacteria. We have also identified heparin binding protein, specific invasion gene, and mammalian cell entry genes. I will show you the operons. There is 44 genes that they are related to infection. So catalase genes, there is 2 superoxide dismutase genes that resist to infection by macrophages. There is CDR4 genes, hemolysin, and the specific lipopolysaccharide coding enzymes, and also some other mycolic acid synthesis protein. And second thing was that we like to understand what is capacity of production of the secondary metabolites, and we analyze the genome, and here are results. So yesterday I talked about non-ribosomal peptide synthetase, and here's genes from these organisms, and the composition of the domains in these genes. Here is name of the genes, the size, and these are different composition of the protein that can specifically synthesize specific product. And on this side, you can see the putative structure that could be synthesized by this product of these genes of non-ribosomal peptide synthetase in nocardia. So here is very interesting one, epoxide compound that could be inhibitor of proteasome in eukaryotic cells. So yeah, this is the specific candidate operon that has 3 genes together, and 71, 72, 73, they are together, and when we analyze structure, we can predict the final structure that is shown here. Okay. And we think that this is specific protein compound that has epoxy structure that could be involved in in biosynthesis of this compound. And this is some okay, so we can see here specific incorporation of the substrate, and at the end we have integral molecules that is shown here. We can identify the molecular weight of this by specific mass spectrometry. And here he's in the middle is a PhD student that he made his PhD on this subject with nocardia. So I will just go further. This analysis of the genome that when we started to work on this, we estimate the size of the genome because at the beginning is always we ask about cost, how much it will cost, how big is genome, and then you say, okay, it will cost I don't know how many euros per nucleotide. So they always ask the size of the genome to accept or reject the project for the sequencing. And I just show this to tell you how these methods without sequencing, they are very useful. We estimate the size 6.2. It is really precise, quite precise. And finally it was 6.1, and I don't know. But so this method is still precise, and I'm really happy to have at this time before sequencing estimation of the genome. When we analyze whole genome, we can see that genes are related to different function, and here you can see different distribution of the genes involved in metabolism, information storage, and processing. Then we can see comparison with some other bacteria, Nocardia farcinica, that infect skin. And what is known that genes are quite at the same position in the chromosome. There is some transposition in comparison of two genomes. So what is interesting that Nocardia farcinica can infect skin, but is not disseminating. Nocardia syria-georgica, it can disseminate in different parts of the bodies. So here is some protein gel of the proteins that are secreted by Nocardia. So some of them, they are toxic for the cells, eukaryotic cells. Some of them we identified by classical identification. The most abundant protein in Actinobacteria in general, if you look secreted protein, this is protein number 1. This is superoxide dismutase. If you have Actinobacteria somewhere, you can add a little bit of the hydrogen peroxide, you will see the bubbles, and this is due to activity of superoxide dismutase, and this is also a specific antigen. When we try to use serum from the mouse, we saw all these proteins. This means that they are secreted in the host, And we used also some human serum from the people with Parkinson's disease. And we got the same response. People with Parkinson's disease, they have a very specific response to this protein. This protein is specific for invasion. So this is fibronectin binding protein. So this protein is very, very immunogenic, and here is just the results of identification of this protein. Yeah. So now we found a lot of proteins secreted in the media, and then we try to understand which system of secretion is present. So we identified a specific twin arginine translocation pathway. It's specific for secretion. Then SEC pathway is known, and also the specific secretion system identified in Mycobacterium tuberculosis. And Nocardia has the same system of secretion. So all these proteins are secreted quite early in the culture, and we can identify that they play a very important role in infection. So this is some cartoon that I try to integrate this early secretion system in Nocardia. Is written here with all components, including the membrane and so on. Here is operon that is important for biosynthesis of a cord factor, that is trehalose dimicolate. So this we identified in the genome. And here's mammalian cell entry operons distributed. I compared with different actinomyces. So there is in Nocardia syriacis georgica, we have 7 clusters of these operons that they are important for degradation, cholesterol, aromatic compounds, and also present in Mycobacterium tuberculosis, Nocardia farcinica, and in Streptomyces cellicolor we have only one operon. So we think that Nocardia syriacis georgica can degrade more complex compounds in comparison with other Actinobacteria. Okay, we found also other gene, and we found also proteasome. So bacteria itself, it has his own proteasome. So it is not same proteasome like in eukaryotic, but it's we think that it evolved to eukaryotics, but Actinobacteria has his own proteasome to remove, and it is resistant to his own inhibitor of proteasome. Okay, what is interesting here is that Nocardia syriacis georgica has 29 P450 genes. So it can use some radical chemistry to destroy, to metabolize some organic compounds. So 29, 27 farcinica, tuberculosis 20. So this makes this bacteria very resistant to antibiotics. Okay, what is surprise? We found rubisco in nocardia. We don't know what does it mean. And finally, I will finish my talk with the conclusion that was made by Blaine Beeman. When whole genome sequence of nocardia asteroides GH2 will be known, we will start new era in research of this black box. So this was a black box for many years. Now we know many things. And I will finish with the name of all these people working on this project. What is said that Blaine Beeman got many times infection with the GH2 bacteria. And what is said that he died from Parkinson's disease. So this is a sad story. And Lovell, his wife, she died also not from Parkinson's disease because Blaine, he explained us that before he piped this culture with with mouth, and several times he drink culture of the nocardia. So that's it. If you have questions, I will be 29. I'm fine. Okay. So we think that during the life, we can be infected many times with nocardia, and little by little our brain start to be depleted with dopaminergic neurons, and with time we develop Parkinson's disease. But Parkinson's disease is a result of the degradation of dopaminergic cells, and many way can produce this phenotype. So one of them is nocardia infection, but we can have also this by chemicals, by some genetic predisposition. We know in some Greek people from Greece, they have a familiary Parkinsonism developed early, since 40 years old, they develop Parkinson symptoms.
Peter,
yes.
Can I didn't catch very well the idea, but how does the nocardia cross the brain-blood barrier?
This is we think that macrophages they start to react with, and then there is lysosome just envelop this bacteria, but it starts to be resistant because cord factor is very resistant to
macrophage
to protect the bacteria. It change form. We know that there is a form like L forms from filamentous to become cocky, and we think that with immunological system it can pass the brain barrier. So
it's helped by the immunology.
Like endobacteria in some immunological system, it can pass.
So can be isolated, or should be growing inside the cell, nocardia?
Yeah, you can it
isolate.
In macrophages it can stay. You can cultivate. You apply on the culture of macrophages, and it will be inside. And there is like Mycobacterium tuberculosis, you can see expression of the gene inside of the macrophages. Yeah, we didn't do this, but is possible. You made a culture of macrophages, you infect, you wait, I don't know, maybe 24 hours, and then you isolate everything. You do dual RNA-seq. You see which genes are important for resistance. We know that, for example, for Mycobacterium tuberculosis, when it is infect the macrophages, they do this dual RNA-seq. They found that Mycobacterium resist due to glyoxylate cycle. It's it's in live, but it use not Krebs cycle because it needs oxygen. It use glyoxylate cycle to survive for many years.
Oops. So I understand that bacteria is going inside the host by breathing, no? The people should breathe something and
in dust, or it could be injured. Yes.
So it seems to have a very neurotrophic behavior because it's
they are specific for dopamine receptors, cells having dopamine receptors. Yeah, and they go to substantia nigra pars compacta, and then they destroy the dopaminergic receptor, yes, or cells.
So I remember your first slide seems to be the nocardia die in a time.
Yes. So immunological system recover from
okay. This cycle response. Or?
I mean, you mean this one?
This one.
Yeah. When some of the mouse will die because they got the infection, but the mouse that survive, they have no nocardia, but they develop this phenotype.
Okay.
Yeah.
So this is a philosophical question, I think. But which should be the opposite of nocardia if finally die in the host? It seems to be not very successful living inside the hostage.
In fact, nocardia is soil bacteria, and this is not some opportunic pathogen. It's not closely related to human. We know that people that they have cancer or they are immunosuppressed, they develop quite often infection by nocardia. We are quite resistant to infection.
If they are very active in recognizing dopamine receptors, should be, I think it affects just the mammals with the...
Yes. We when I was on my postdoc, we identified something like 30 sea mammals infected with nocardia from SeaWorld, from Los Angeles. They send us the samples from the killer whale and from foca and we identified nocardia. They are often infected with this bacteria, with nocardia. This is a main problem for the health of these sea mammals in captivity. But there is also oysters. There is fish that they have some nocardia salmonicida and so on. So not only this species of nocardia that infect Otidis caviarum and so on. Yeah.
I'm not a neurologist, but I think that dopamine receptors are only located just in the brain, not in the lung, for example, or
they are mostly in the brain. Yeah.
Some question.
Okay, so I don't know who is behind now. I should stop. If you have questions, maybe we can discuss later. Thank you very much for your attention.
No le hago, bro. Can I ask you a question?
Yeah.
Yes. I don't remember if you said something about it, but about the treatments.
Yeah.
Or is there a treatment? Because
yes, there is a there is a treatment for this bacteria. Is amikacin, is some aminoglycoside antibiotic. Majority of nocardia are resistant to beta-lactam antibiotics, and cycloserine is one of the choice, and trimethoprim, and amikacin is antibiotic of choice.
Okay.
And there is different. For example, the drugs against the Mycobacterium tuberculosis are not effective on nocardia.
Okay.
And it's quite difficult to treat these infections, because once a person is immunocompromised, like after treatment with for the cancer treatment, chemotherapy, they became very sensitive to infection. Also, people that got organ transplantation, they are very, very susceptible to infections of nocardia. They must be very, very careful. And usually, medical doctors, they forgot about this infection, but usually in Lyon we have Center for Nocardia Research and big bank of the nocardia, and they send always samples to ask lab, could you confirm if it is nocardia or not?
So if someone has Parkinson, maybe if I have Parkinson, I have to go test to see if has a chemical or nocardias.
Yeah, there is unfortunately confirmation of Parkinson disease is post-mortem. You you confirm this by pathologist looking these Levy bodies, and then we can tell, okay, you got. But of course, we can predict if you have some movement problems and so on. We can tell, okay, it is probably Parkinson. But the final diagnosis of the Parkinson is Levy bodies in the brain.
But I don't understand how the treatment works if you know that you had Parkinson when you die. How do you know you have this kind of Parkinson caused by nocardia?
No. If you have Parkinson, you have some problem of movement. So there is drugs, L-dopa, carbidopa.
Okay, I understand.
Yeah, but these are not related to this is for depletion of dopaminergic neurons, and not to infection. But if you got infection, you have fever, like other infections, and then doctor give you one antibiotics, you still have fever, and then they try to isolate bacteria and to see if it is, I don't know, nocardia or some other bacteria, and then they do also antibiogram, and they say, okay, this is nocardia, you should apply some specific antibiotics according to the antibiogram.
Okay, thank you.
Okay.
Just one more question. Okay. I'm just connecting with some paper that I have written. Dopamine should be might be recognized by some other bacteria in a microbiome, human microbiome. I read some paper. People are saying has dopamine is working as an autoinducer for quorum sensing gram-negative behavior. So it's very huge field of research by now. dopamine connection with the microbes.
Sure.
So Petar, again, thank you very much for your nice presentation.
It was my
we need to
pleasure.
have Oh, Gabi, are you ready? No. Give me. Now you'll be here with us.
Yeah. Oh, yeah.
Gabi. Petar, I would like to introduce myself. Very nice to meet you. Ya. ¿Qué? No hay problema. Todo bien. Les voy a dejar con mi estimada colega. Sí. Claro, sí. Ahora sí. No, el micrófono quiero presentar a Gabriela, nuestra colega que ha formado parte también del grupo. Gabriela Sánchez, que es profesora asociada de la Facultad de Microbial, nos va a mostrar su enfoque, su mirada investigadora acerca de las biopelículas en el ecosistema. Así que Gabriela, te agradecemos el haber venido y queda a disposición completa del auditorio.
Muchas gracias por estar aquí. Agradezco al profesor Homero en esta invitación de participar junto a todo su equipo en un hermoso Fondesit que están realizando. La verdad es que quiero hacer una pequeña contribución. I hope that you can understand me, because my English is so poor. I speak in Spanish. Mi pequeña contribución a lo que tiene que ver con la microbiología, ¿cierto? Desde el punto de vista clínico. Así que llamamos a esta presentación Oral Biofilms and Oral Microbiome from Dental Ecology to Systemic Health. Yo soy odontóloga desde el año 2005 y después de eso ingresé como cualquier dentista graduado a trabajar en el sistema público, a atender pacientes. Y por esas cosas de la vida fui llamada a la academia y inicié este camino de formación como docente primero, mi magíster es en educación, y luego me picó el bichito de hacer un doctorado, como muchos de los que están aquí, y chiquillos se pueden. Y mi doctorado fue en la Facultad de Ciencias Biológicas, en el doctorado de microbiología, y estoy muy contenta porque aprendí mucho y la verdad es que si bien de formación soy odontóloga, yo creo que mi corazón es algo de microbióloga. Quiero agradecer también al Fondesit de Iniciación, que ya culminó el 2023, pero me ha dado las herramientas para poder generar investigación de buen nivel en nuestra facultad, financiada. Ustedes saben que es muy caro hacer investigación, y hoy estamos actualmente haciendo un proyecto donde soy la directora general de este proyecto de inserción en la academia, donde una ingeniera, Camila Quesada, se ha insertado en la Facultad de Odontología para hacer un fortalecimiento en la investigación, sobre todo partiendo desde el diagnóstico y el tratamiento centrado en pacientes de patologías orales complejas. Cuando pensamos en microbioma oral, nunca se nos imagina que es esta boca hermosa. Uno pareciera que dijera, esta boca es estéril, ¿cierto? No tiene microorganismos, está hermosa. Pero la realidad de nuestros pacientes, de nosotros mismos, ¿cierto? Dista mucho de eso. Y hay una hermosa biopelícula organizada, ¿cierto? En nuestras superficies, tanto dentarias, las mucosas, y que tenemos que mantenerlas en un estado, ¿cierto?, de eubiosis, porque si ellas se descontrolan, generan un proceso de desequilibrio y finalmente establecen patología, ¿ya? En ese sentido, se ha descrito que la boca tiene muchos nichos ecológicos y da cabida a muchos microorganismos. Pensando en el nombre de este simposio de Actinobacteria, ¿cierto? La verdad es que yo decía, pucha, nuestro grupo es más de otros, ¿cierto? Géneros no tan parecidos a las actinobacterias, pero sí, ellas tienen un rol dentro de lo que puede visualizar bastante fundamental en el equilibrio de esta microbiota y todos sus genes concatenados, ¿cierto?, en lo que tienen que ver con la boca. Entonces ahí tenemos la placa supragingival, la mucosa bucal, la mucosa del dorso de la lengua, y son muy ricas en microorganismos. Y ahí aparecen, ¿cierto?, sobre todo en el dorso de la lengua, nuestros queridos actinomyces, que le dan el nombre de su grupo, ¿cierto?, a el filum Actinobacterias. Sin embargo, cuando comenzamos a trabajar en microbiología, uno tiende a hablar de bacterias y sabemos que la microbiota está formada por otros componentes además de los bacterianos, ¿cierto? Y aquí, por ejemplo, claramente se describen hongos que habitan y cohabitan con el grupo, ¿cierto?, con las bacterias en nuestra cavidad oral. Y saliendo de ello también tenemos las arqueas. Y pensando que en nuestro conocimiento microbiano de arqueas sabemos que son bacterias que nosotros le llamamos súper extremófilas, ¿cierto?, que son a todo combate. Eso quiere decir que nuestra cavidad oral realmente ofrece muchos desafíos a nuestros microorganismos que conviven con nosotros. Pensemos que desde nuestro café calentito de casi 50 grados Celsius en la mañana a tomarse un helado así casi de 2 grados, estos microorganismos tienen que jugar en distintas condiciones, pHs, nuestros alimentos, nuestros hábitos de fumar, de beber, ¿cierto? Hay distintas condiciones en que nuestros microorganismos tienen que jugar ciertos roles adaptativos para poder ganarle a estos nichos ecológicos y competir entre ellos. Entonces las arqueas no están ausentes de la microbiota oral humana. Se ha descrito también los virus que también conviven en cavidad oral, y claramente se diferencian los virus presentes en salud y en enfermedad. Por lo tanto, algo también ellos tienen que decir en este conglomerado, ¿cierto?, microbiano. Entonces, esta microbiota que parte desde la boca, por eso es tan fundamental, porque el microbioma humano está desde la boca, ¿cierto?, hasta el ano y algunas de nuestras mucosas, ¿cierto?, como la piel o las de las zonas de nuestro sistema reproductor, nos acompaña a lo largo de toda la vida y va sufriendo ciertos cambios propios etarios, pero también tienen que ver con los cambios que tienen que ver con los factores de cómo nacemos, si nacemos por vía vaginal o por vía cesárea, ¿cierto? Nuestra alimentación inicial, si fue de leche, ¿cierto?, materna o leche de fórmula. Y luego todo el componente genético también, por supuesto, que se suma a los ambientales. Nuestra higiene oral, nuestra dieta, el estrés, nuestros hábitos, ya sean buenos o no buenos, ¿cierto? Y esto va a influir claramente en nuestro sistema inmunológico y por lo tanto las condiciones de salud ya en las cuales nosotros nos enfrentamos. Así es que nuestro microbioma oral, al sufrir un desequilibrio, va a generar patología en distintos nichos, ya sea a nivel gingival, a nivel periimplantario, cuando estamos ya, ¿cierto?, con algún aparato protésico a nivel bucal, a nivel apical de los dientes, cuando los dientes se ven, ¿cierto?, inflamados dentro de sus tejidos, a través de caries, y todo eso afecta, ¿por qué? Porque los microorganismos de nuestra microbiota normal, que son nuestros comensales y nos ayudan a mantener esta homeostasis en general, también se vuelven oportunistas, patógenos oportunistas que finalmente van a generar patologías. Está claramente descrito, ¿cierto?, que nosotros tenemos que propender a una microbiota bien eubiótica, ¿cierto?, que tenga interacciones cooperativas, pero que siempre van a estar presentes estos keystone pathogens, ¿cierto?, que son patógenos oportunistas, que son importantes en el establecimiento de una microbiota disbiótica y finalmente lo que se traduce en pérdida de tejidos de soporte y finalmente pérdida de tejido o de piezas dentarias. Ya hay gran avance en la literatura de demostrar que nosotros podemos darle condiciones a nuestra microbiota para que sea una microbiota eubiótica. Y en ese sentido, el potencial, por ejemplo, prebiótico que tienen los nitratos es altamente deseable porque genera un ambiente de pH adecuado para nuestra boca y eso de alguna manera se traduce en condiciones que favorecen tanto la homeostasia a nivel oral, pero también a nivel corporal. Y les voy a demostrar un poquito más adelante acerca de ello. Se sabe que hay géneros que son reductores de nitrato y eso va a ser asociado a la condición de salud. Esa condición de aumentar o estabilizar el pH, cuando el pH es más ácido, siempre es más dañino para nosotros, ¿cierto? Estabilizamos un pH adecuado y nuestra boca se genera como una especie de motor de nuestro sistema inmunológico en general. Ya aquí está claramente demostrado, ¿cierto?, que la microbiota se va a hacer cargo, dependiendo de las vías metabólicas, de desarrollar su metabolismo alrededor de carbohidratos, de proteínas, pero también de algunos compuestos desde los vegetales o de, por ejemplo, las carnes. Y principalmente los vegetales de hoja verde se ha visto que están en esta vía, ¿cierto?, de el desarrollo de sintetizar o de partículas o su metabolismo a partir del cambio de los nitratos a los nitritos, y eso claramente disminuye, ¿cierto?, todo lo que es el estrés, mejora la homeostasis de la presión sanguínea, son antiinflamatorios y propenden a un estado de salud corporal, a diferencia de otros compuestos que son, ¿cierto?, otros microorganismos que están en otras vías metabólicas en los cuales aumenta, ¿cierto?, también la homeostasis de la sangre, la antiinflamación, a diferencia de en otras vías en las cuales aumenta la periodontitis, ¿cierto?, aumenta la inflamación, y también se ha visto en relación a problemas de hiperactividad, inflamación y agentes que son más bien tendientes a patologías cardiorrespiratorias. Entonces, eso es lo que mencioné ahí, que Actinomyces está en el grupo de los buenos, así que ahí también me sumo al nombre de nuestro simposio, ¿cierto? Entonces, también esta diapositiva, alguna vez se la robé al profe Homero cuando hizo una clase que es del año, yo el doctorado lo hice hace hartos años atrás, prácticamente como 13 años atrás, y él nos mostraba cuáles eran las etapas de la formación de la biopelícula, y lo más importante, lo que más me gusta de esta diapositiva es su referencia, que es el profesor Costerton, que es un dentista y que ha generado toda esta teoría de la formación de la biopelícula que ha servido para todo el campo clínico, médico, ambiental, porque en todas partes donde conocemos que se forman las biopelículas pasan por las mismas etapas. Y de hecho el profesor Concepción en algún momento vino a Chile, estuvo acá con el profesor Homero, lamentablemente él falleció de forma un poco inesperada, un poco antes de lo que se esperaba. Pudo haber sido mi cotutor de tesis, pero bueno, la vida dijo otra cosa y hacemos el honor a esta mención de este gran dentista que generó esta descripción clara de la etapa importante de la formación de las biopelículas, donde sabemos que nuestros microorganismos, alrededor de su 80-90%, están asociados en biopelículas más que de forma planctónica. En la cavidad oral sabemos que hay colonizadores primarios, ¿cierto? Y colonizadores más bien tardíos, donde se van generando condiciones de cohabitabilidad, también competencia, ¿cierto? Y aquí aparecen varias de las líneas de los actinomyces en aquellos colonizadores intermedios, Actinomyces oralis, Naeslundi, ¿cierto? Y aquí israeli, que forman parte de nuestra microbiota oral y que de alguna manera necesitamos dar condiciones, ¿cierto?, para que generen una eubiosis de la microbiota y no una disbiosis de ella. Este mismo trabajo menciona sobre, ¿cierto?, estas etapas de formación. Tenemos las etapas iniciales, por eso es tan importante las patologías o digamos los controles de la microbiota a nivel oral con nuestras técnicas de cepillado, porque finalmente manejamos nuestra microbiota en estas etapas, evitando, ¿cierto?, esta transición a que haya un crecimiento y una colonización de microorganismos que pueden ser potencialmente patógenos, ¿ya? Pero sabemos que la intercomunicación dentro de las biopelículas es multiradial, hay cierta protección de antimicrobianos, se generan transferencias de genes horizontal, ¿cierto? Hay una cohesión, una organización, también las vías metabólicas a partir de los alimentos que, ¿cierto?, el hospedero les ofrece, y todo ocurre en esta superficie de biopelícula adherida a las superficies orales que pueden ser dientes o mucosas. Se ha descrito en relación a patologías de caries, por ejemplo, que la microbiota asociada a lesiones de caries superficiales o de fosas, ¿cierto? Son totalmente distintas a aquellas que son más profundas. El comportamiento y la composición de estos microorganismos va cambiando a medida que se van introduciendo los túbulos dentinarios, van a zonas mucho más bajas en tensión de oxígeno, baja en lo que es la adherencia a los metabolismos sacarolíticos y se colocan más bien proteolíticos debido a que su fuente de alimento principal ya pasa a ser, ¿cierto?, todo el componente biopulpar y sanguíneo que hay dentro de los dientes. ¿Ya? Y en esta diversidad, ¿cierto?, es que los microorganismos, algunos de ellos más bien con capacidades patogénicas, ¿cierto?, se van estableciendo, se van adheriendo, principalmente, por ejemplo, muy descrito en caries Streptococcus mutan, este productor de ácido, ¿cierto?, y que genera la disolución del tejido duro del diente y que finalmente permite la invasión de otros microorganismos que son, de punto de vista más oportunistas, ¿cierto?, para introducirse hacia los túbulos de la zona más profunda, y es así como va madurando, ¿cierto?, esta biopelícula para convertirse en un ambiente muy ácido y muy patogénico, en definitiva, sobre todo porque sus productos migran hacia la zona de la pulpa dentaria y finalmente producen la necrosis pulpar. Otras patologías asociadas a los tejidos de soporte, por ejemplo, la periodontitis marginal, ¿cierto?, donde estos microorganismos se alojan ahí en el surco supra y subgingival. También van cambiando la composición porque la tensión de oxígeno va disminuyendo a medida que nos vamos profundizando los surcos periodontales, y lo que hacen estas bacterias es disromper, ¿cierto?, el epitelio de unión y finalmente migrar a zonas mucho más profundas, prácticamente anaerobias, y efectivamente le dan una condición más bien patológica a esta microbiota. En periimplantitis no es la excepción. Lamentablemente en algún tiempo se describió que la implantología a nivel oral iba a ser como la panacea, no importa, sácate los dientes porque te podemos colocar un implante. Sabemos que el implante primero no se coloca en un ambiente aséptico, está la saliva, están los microorganismos ahí presentes cuando se hace el lecho donde se inserta el tornillo, ¿cierto?, del implante, y luego de ello hay colonizadores también primarios y tardíos, y esto no se controla. La patología, si bien no es igual a la periodontitis, pero es muy similar, aparece una mucositis, una inflamación de la mucosa y finalmente la pérdida del implante por pérdida del hueso, y no hay una unión, ¿cierto?, del implante hacia el hueso, que es lo que se espera cuando el implante funciona de manera adecuada. Y finalmente la periodontitis apical, que se deriva en gran parte, en gran medida, por la caries, que ya se los había mencionado hace unas diapositivas anteriores, pero hay otras vías de infección, por ejemplo, en trauma. Nosotros tenemos una alta prevalencia de trauma, ya sea asociado al deporte, ya sea asociado, ¿cierto?, a el grado de violencia que hoy en día nuestra sociedad sufre, ¿cierto? Los pacientes llegan traumatizados con exposición a nivel de la pulpa dentaria que debería mantenerse aséptica en condiciones de salud. Los microorganismos invaden y finalmente lo que se produce, ¿cierto?, es una necrosis del tejido pulpodentinario de la pulpa, ¿cierto?, que es un paquete vasculo nervioso, y que finalmente genera destrucción del hueso a nivel apical, es decir, de la punta de los dientes. Se han descrito otras vías, por ejemplo, la hematógena, pero las más importantes son estas tres. Y en ese sentido, sabemos que el diente, ¿cierto?, en su estructura, este tejido duro, que incluso a veces es más duro que el mismo hueso, ¿cierto?, está formado tanto por esmalte en la parte externa como por la dentina en la parte interna, ¿cierto?, y la pulpa, que es este paquete vasculo nervioso que le da el soporte de vida, ¿cierto?, le da nutrición, le da seguridad a este órgano que es el complejo pulpodentinario. Sin embargo, como les dije, el trauma, la caries, anomalías dentarias también van a hacer que estos microorganismos que eran de nuestra microbiota oral invadan, ¿cierto?, y se transformen en patógenos oportunistas, generando la patología apical. Ahora, ¿qué ocurre? Que estos microorganismos no entran planctónicamente. Nuestra anatomía del tejido dentario tiene túbulos que son de alrededor 5 micrones, que por lo tanto le da una cavidad inmensa a estos microorganismos que son mucho más pequeñitos, ¿cierto? Y generan infecciones primarias, secundarias, persistentes y además extrarradiculares en la zona externa al diente. Todo esto organizando, ¿cierto?, una biopelícula que forma la periodontitis apical y que es la que yo me he dedicado a estudiar porque finalmente mi especialidad odontológica es ser endodoncista. Yo gran parte de lo que hago durante el día, de hecho hoy día vengo con mi nariz un poco marcada porque uso magnificación y eso genera un aumento en la visión en lo que estoy buscando, ¿cierto? Y ese es mi diario vivir, trabajo mucho de esto diariamente, me encanta estar en el laboratorio, pero mucho de lo que hago también forma parte de la atención de pacientes y tratando la periodontitis apical. Durante el desarrollo de mi tesis de doctorado, el año 2016 más o menos estaba la estructura y yo quería investigar la periodontitis apical asociada a fracasos de los tratamientos endodónticos. Y en ese tiempo era el boom, ¿cierto?, de todo lo que eran las tecnologías de secuenciación. Y la verdad es que si bien en otras áreas de la salud, en otras áreas del punto de vista ambiental, ecológico, la secuenciación ya llevaba harto tiempo en el campo, la verdad es que para mi área en endodoncia, solo a esa fecha habían 6 artículos publicados con secuenciación genómica, ya utilizando principalmente técnicas de pirosecuenciación y 2 de ellas con tecnología Illumina. ¿Ya? Y todos ellos eran de poblaciones principalmente norteamericanas, asiáticas, europeas, y solo una, que era del doctor Siqueira, era en población brasileña. ¿Qué nos mostraba el paper del profesor Siqueira? Es que la composición de esta patología, ¿cierto?, persistente, formaba gran parte de distintas filas, ¿cierto? Pero además nos mostraba un gran componente de bacterias que estaban asociados a la atención en salud. Era una contaminación intratratamiento, porque las pseudomonas, principalmente las pseudomonas que fueron pesquisadas en el 100% de los pacientes, forman parte de nuestra microbiota normal. Y estos dientes que fueron estudiados, no había forma de que ese microorganismo entrara a la boca si no fue posterior o durante el tratamiento endodóntico. Posterior en ningún caso porque la restauración estaba en forma adecuada, por lo tanto la hipótesis era que esto había sido contaminación intratratamiento, problemas en la desinfección de los mismos clínicos que realizaban el procedimiento. ¿Ya? Y basado en eso nosotros avanzamos en hacer un estudio que en Chile fue muy bien recibido y también tuve la colaboración de colegas de acá de biotecnología, el profesor Gallardo y su grupo, ¿cierto? Donde finalmente me ayudaron a entender estas tecnologías que yo realmente para mí escapaba mucho en mi entendimiento de cómo aplicarlas. Recibí mucha ayuda, tomamos la pesquisa de 24 pacientes con este diagnóstico y hicimos algo que no habían hecho los colegas en otros países, que es cruzar la información con datos clínicos, con cómo eran nuestros pacientes, tomando en cuenta si eran hombres o mujeres, la edad que tenían, la condición sistémica, si las lesiones eran dolorosas o no dolorosas, el nivel de pérdida del hueso y así también como el lugar donde estaba colocado el diente, si el tratamiento era adecuado y efectivamente qué data del tratamiento teníamos. Y bajo ese sentido, lo importante o lo más rescatable de esto es que nos dimos cuenta que la microbiota asociada a lesiones persistentes en pacientes con alguna patología sistémica era muchísimo más diversa que aquella que estaba en pacientes con condiciones de salud normal. Y esto obviamente nos hace sentido, nos da cuenta de que efectivamente el sistema inmunológico mantiene a raya cierta diversidad bacteriana en esta zona, a diferencia de aquellos que tienen algún tipo de patología, y hay una clara relación con el estado de salud del paciente. Aquí uno siempre piensa que es primero, ¿cierto? El huevo o la gallina, o es la periodontitis la que genera esto, o es que los pacientes que ya tienen esta condición no logran combatir la periodontitis. Entonces está esa pregunta abierta, obviamente. Y bajo ese sentido empezamos a pensar, ya, si el colega efectivamente, tenemos que pensar que los colegas hacen todo su mejor esfuerzo, entonces finalmente nuestras estrategias de desinfección ya no están siendo adecuadas, por ejemplo, por resistencia, el uso y abuso del mismo medicamento o los tratamientos de desinfección y de antisepsia de nuestras patologías no están dando efecto. Por ejemplo, en el caso de algunos enjuagues bucales que nosotros también utilizamos en la desinfección, ¿qué hacen? ¿Son efectivos, no son efectivos? ¿Son buenos o no buenos? ¿A qué propenden? ¿A generar una disbiosis en el microbioma o a un microbioma eubiótico. Y la verdad que, por ejemplo, en relación a la clorexidina, está claramente demostrado que la clorexidina genera un desequilibrio en la microbiota humana oral. Ya genera condiciones de disbiosis bacteriana, hay desequilibrio y por lo tanto podríamos, pensando en bajar la cantidad, ¿cierto?, de microorganismos, dejando microorganismos que son potencialmente patógenos para nuestra boca. ¿Ya? Entonces, en ese contexto es que la segunda parte de esta tesis doctoral y que finalmente se tradujo en el Fondesit de iniciación es buscar alternativas de antisépticos bucales para no impactar desde el punto de vista de la eubiosis de nuestra microbiota y comenzamos a pensar en las nanopartículas de cobre, ¿ya? Las nanopartículas en general han sido moléculas utilizadas en varios campos de la odontología como antisépticos, ¿ya? Porque controlan los microorganismos y lo bueno que tienen en general las nanopartículas, sobre todo también las de cobre, es que tienen varios targets de acción sobre las bacterias y por lo tanto es muy difícil que generen una resistencia, porque pudiesen generar resistencia en uno de los puntos de target, pero los otros todavía siguen siendo susceptibles. Por lo tanto, podrían tener una actividad, ¿cierto?, antibiopelícula. Y como les decía, el cobre o las nanopartículas de cobre ya se había reportado, por ejemplo, para hacer brackets de ortodoncia, algunos enjuagues bucales, algunos materiales, por ejemplo, para relleno de restauraciones, en implantes dentales, etc. Había una serie de utilizaciones, pero la mayoría de ellos decía cierto concernimiento en relación a la citotoxicidad. Es por eso que nos propusimos en el Fondesit hacer una síntesis de una presentación de una nanopartícula funcionalizada que tuviera tanto el potencial antibacteriano, pero que además fuera biocompatible, es decir, que evitara lo que se estaba reportando en la literatura de esta toxicidad de la nanopartícula de cobre. Y es que hicimos una metodología junto con los estudiantes de ingeniería, del doctorado de Ingeniería de Materiales, en lo cual mezclamos plata y cobre y logramos sintetizar una propuesta, ¿cierto?, de un gel de nanopartículas, ¿cierto?, que fuera tanto biocompatible como antibacteriano, y ahí hicimos análisis también de citotoxicidad. Ese trabajo lo publicamos en este artículo, que es una de las revistas más importantes de la odontología, si bien yo sé que a nivel de la investigación humana, yo siempre digo a mis estudiantes, miren, el journal, digamos, el área de odontología del Web of Science tiene alrededor de 170 papers que son reconocidos por la Web of Science. Y el más grande tiene un nivel de impacto no más allá de 12 o 13. Y ustedes imaginarán que otras revistas pueden tener 50, 60, 70, somos chiquititos en el área odontológica, pero de alguna manera donde hemos podido presentar nuestros resultados en los top 10 de los journals en odontología, y eso nos llena de bastante orgullo. Así que este trabajo lo presentamos presentando este gel, ¿cierto?, desinfectante, la síntesis de él para poder controlar, y demostrábamos, ¿cierto?, su actividad antibacteriana, sobre todo de una de las proposiciones, porque probamos varias de las posibles estrategias para poder quedarnos con una presentación que finalmente este trabajo nos dio incluso un premio por la Sociedad Americana de Endodoncia como el mejor paper reconocido dentro del Basic Research Technology. Es uno dentro de muchísimos que se publican al año y la verdad es que también estamos muy contentos por este reconocimiento. Este trabajo lo patentamos, lo presentamos a patente y ya está concedida la patente, así que seguimos trabajando en esa línea porque nuestra idea es seguir demostrando. Nosotros estos tipos de compuestos no puedo llegar y hacer un ensayo clínico en paciente, tengo que recibir todas las aceptaciones, ¿cierto?, y los permisos que tienen que ver con el ISP y las normas para poder probarlos en pacientes. Hicimos un primer estudio en un modelo animal pequeñito, ya que es una ratita spray dolly que tenemos acá, ¿cierto?, en el bioterio de la universidad, donde también lo utilizamos, si bien no lo pudimos usar dentro de los dientes, porque los dientes de los ratoncitos, si ya el ratoncito es chico, el diente del ratón es enanísimo, era muy difícil hacer el modelo, pero al menos lo usamos como desinfectante oral, ¿ya? Así que le lavábamos la boquita al pacientito, digo yo, por varios días, algunos hasta por 30 días, y logramos demostrar que al menos nanopartículas por 7 días su comportamiento era bastante adecuado. Hicimos también nuevamente, ¿cierto?, tecnología de nueva secuenciación para poder establecer las comunidades bacterianas, cómo se veían afectadas, y además hicimos todo este análisis de citotoxicidad presente en órganos nobles. Y finalmente lo que pudimos encontrar como resultado es que al compararlo con clorhexidina, ¿cierto?, nuestras nanopartículas de gel aumentaban la riqueza, aumentaban la diversidad, generaba estas vías funcionales de los nitratos a los nitritos, y eso nos dejó bastante contentos que aparecieran, que potenciaban estos microorganismos que generaban estas vías metabólicas. Así que nuevamente vamos hacia la búsqueda de generar condiciones más eubióticas con los desinfectantes orales. Esto tiene mucho camino por recorrer todavía. En este estudio en animal pequeño, ¿cierto?, pudimos ver su estabilidad fisiológica, la modulación a nivel sanguínea, y que los parámetros bioquímicos se mantenían dentro de estado, y no vimos bioacumulación tampoco dentro de los tejidos, pero necesitamos escalarlo a cosas más grandes. Así que presentamos este proyecto el año pasado para el Fondef-IDEA, que salió ahora en junio, julio, que tenía este nombre de un gel antiséptico endoperio perimplantario para ya una aplicación más bien odontológica, para poder obtener ya las últimas pruebas necesarias para poder tener los permisos del ISP y hacer algún estudio en pacientes. Sin embargo, así es la investigación, chiquillos, y por eso vengo a motivarlo. Hubo 102 proyectos financiados de las 871 postulaciones, y nuestro trabajo quedó en el primero en lista de espera. Así de motivante. Y empaté con el último que quedó. Y no sé por qué fue la característica, por qué empatamos y a mí me dejaron afuera y a ellos adentro. Parece que tiene que ver algo también con equidad de género. Nosotros íbamos 2 hombres, 2 mujeres, pero también ven cuántos hombres o cuántas mujeres se han adjudicado. Y a veces antes favorecía a la mujer, ahora es como bien equilibrado, y me parece. Así que me dice que estoy en lista de espera al menos hasta cuando se inicie el otro Fondef, así que podría pasar cualquier cosa en algún proyecto que se cayera. No le quiero desear mal a los colegas que se lo ganaron, pero en una de esas. Pero si no, ya estoy trabajando para presentarlo nuevamente, porque creo que tiene potencial para seguir trabajando. Y finalmente, comentarles que estamos también en estas áreas de nanobiosensores. Esta cosa de trabajar con los ingenieros, con las nanotecnologías, me permitió estar en el claustro de ingeniería material y trabajar con 2 estupendos estudiantes que ahora ya son doctores. Uno es Camila Quesada, que fue quien se insertó en la academia con nosotros en la Facultad de Odontología, que ella desarrolló un sensor de nanopartículas impresas como para sintetizar finalmente imagínense cuando uno hace test de ELISA, en los cuales hay anticuerpos, ¿cierto?, que son muy lábiles y hay que tenerlos congelados. Entonces, la idea era hacer estos share inside, es decir, que ojalá el colega que quiera detectar algo pueda hacer una muestra simple con algo que no se desintegre, que dure en el tiempo y que sea altamente sensible. Así que ella generó esta una formulación con nanopartículas impresas para detectar, ¿cierto?, algunos marcadores de cáncer, ya así como una tirita, ¿cierto?, de esto como un ensayo de COVID. Y este otro es de mis estudiantes que también ha trabajado en lo que es nanobiosensores ya, pero ahí ya integrándolos más bien a algo más bien electrónico, también pensado en muestras simples, no sangre, sino que quizás saliva o algo otro marcador. En este caso estamos trabajando con biomarcadores de cáncer, y que nuevamente en el proyecto de Camila lo vamos a retomar haciendo toda esta conexión de la microbiota oral que pudiese estar haciendo un eje, ¿cierto?, boca-colon de la alta prevalencia de colon, de cáncer de colon. Así que en esto también nosotros lo patentamos, mandamos la presentación, estamos a la espera de la resolución de la patente en conjunto con la Universidad de Chile, y presentamos un anillo regular al cual estamos en la espera ya para poder estudiar, ¿cierto?, estas potenciales lesiones malignas orales y como un screening inicial. Así que estimados colegas, como conclusión de esta presentación, quiero mencionarles que a mí me encanta esto y los felicito por estar acá. Yo creo que siempre escuchar a alguien, incluso de un área tan distinta a la que quizás ustedes puedan ser, nos deja mensajes, nos deja herramientas. Yo, al ser profesora de odontología, muchas veces soy el señor Corales, porque barro, canto, corto los tickets, hago todas esas cosas, porque muchos estudiantes de posgrado no tenemos, es decir, son muy pocos los estudiantes de posgrado, y de ahí, por eso me he ido incorporando a otros núcleos, a otros claustros, para poder trabajar con estudiantes que han sido maravillosos los con los que he trabajado, pero esta es mi vida diaria, aquí estoy, siempre las preguntas vienen de la clínica, de los pacientes, del dolor, de la persistencia de las infecciones, y es parte que también me motiva diariamente a dar un mejor servicio. ¿Y por qué las respuestas a las preguntas clínicas tienen que venir del extranjero? Yo creo que Chile está capacitado para levantar mucha hermosa y buena información y buena evidencia de calidad. Así que a eso los motivo, a seguir trabajando por nuestros pacientes. Muchísimas gracias.
Quedamos atentos a las preguntas de los estudiantes de este fascinante mundo del microbioma oral. Por favor.
Primero, muchísimas gracias por la presentación. Importante mencionar que mi campo es completamente distinto. Yo estudio biotecnología vegetal, así que... Pero mi pregunta es sobre las partículas de cobre, ya que no tengo claro si este tratamiento se ocupará para gente que tiene enfermedades dentales o para gente sana también, porque ¿cómo las partículas serían selectivas y no matar a la película buena de la boca?
Sí, como está pensado en una desinfectante con varios usos, en el caso de las enfermedades gingivales o perimplantarias, podría ser como una especie de enjuague o de antiséptico tópico en etapas tempranas, por ejemplo la mucositis o lo que le llamamos nosotros la gingivitis, que son previas a la etapa ya grave en la cual el diente ha perdido tejido de soporte. Entonces, en ese caso, son terapias acotadas de 7 días en general, en la cual tú, pues ya demostramos que en 7 días genera una condición de microorganismos bien eubióticos y que no va a alterar, a diferencia de lo que ahora hacemos, que es colocarle el enjuague de clorexidina, y la clorexidina finalmente igual vemos que genera una condición de comunidad bacteriana más disbiótica. Entonces, esa es como por ese lado. En el caso de la periodontitis apical, que ya vamos a dejar dentro del diente cuando hay una necrosis, cuando tienen que hacerse una endodoncia, ahí también se piensa en una medicación de 7 días para desinfectar el cuadro como agudo y dar condiciones de reparación del tejido que se ha perdido a nivel apical. En general no es como para un enjuague bucal para una paciente sano. Se piensa en tratar patologías, pero en sus etapas tempranas, para poder evitar que esto se perpetúe o genere patologías mayores.
Genial, gracias.
¿Qué tal, doctora? Felicidades por la charla. Primero, increíble el nivel de conocimiento en microbiología en su especialidad. Yo soy hijo de odontólogos, hermana odontóloga y también una tía odontóloga, así que voy a transmitir una de Argentina, ¿no? Y la están viendo desde el norte de Argentina y le voy a transmitir una pregunta que me hicieron, pensando mucho en este control de patógenos. Mi mamá me mandó esta pregunta que dice... si usted tiene idea del uso del Lactobacillus reuteri para como control, digamos, o para desorganizar la placa que queda bajo, que le leo exactamente, que queda bajo la encía, donde nos llega el cepillado, digamos, esta bacteria queda y se protege con el biofilm y es lo que sigue destruyendo hueso alveolar y produce retracción de encía y sobre todo mujeres mayores de 50 años, creo que la bacteria es Porphyromonas gingivalis. Especialmente para estos tratamientos que usted hablaba de colocar ¿cómo se llama? Un implante o como un probiótico. ¿Cómo cree usted que se podría usar?
Sí, lo entiendo. La verdad es que en el área odontológica se describe el uso de varios probióticos que pudiesen propender a ganar este nicho ecológico de los patobiontes o de los keystone pathogen. En ese sentido, uno de los microorganismos que usted menciona, pero hay varios estudios de otros lactobacilares, efectivamente, y todo está tendiendo a buscar estos microorganismos que pudiesen generar vías, ¿cierto?, más bien propendientes a las rutas metabólicas de los nitratos. ¿Ya? En la Universidad de Chile hay un gran grupo de investigación en el cual están trabajando sobre microorganismos que pudiesen cambiar la metabólica de la boca e incluso tienden a decirte come más cosas verdes, sobre todo más que las lechugas, por ejemplo, las que son las hojas de espinaca, ese tipo de... tienen harto nitrato y finalmente le das la condición como la comidita a estos microorganismos y propendes a que ese grupo microbiano sea más que el otro, más que insertarlo en pacientes que ya sean, que tengan, por ejemplo, la detección de Porphyromonas gingivalis. Ahora, lo importante es entender que si bien Porphyromonas gingivalis yo le llamo como el gold estándar en estudios en microbiología de periodoncia, por ejemplo, agregar Actinobacter en periodontitis agresiva, etcétera, tenemos súper claro que en nuestra comunidad es polimicrobiana, es decir, no es solo un microorganismo. Entonces ahí tenemos que jugar, claro, yo ataco a uno, pero ¿qué le voy a estar haciendo al otro? Como la pregunta que hacía ella, ¿cierto? Tenemos que tener un equilibrio súper adecuado para no afectar a los que realmente pudiesen ser potenciales probióticos de nuestra boca. Así que hay varias posibilidades en relación a los probióticos. Yo sé que, bueno, aquí ustedes en la Facultad de Ciencias Biológicas, la profesora Pola tiene claridad en eso y en algún momento también ha leído, ¿cierto?, acerca de los probióticos a nivel oral, que es la puerta de entrada, eso es lo que decíamos. De repente hay desequilibrios acá, pero cómo están influyendo a nivel de la mucosa gastrointestinal, que también es otro mundo.
¿Y usted cree que estos tipos de probióticos podría ser utilizado como un tratamiento pre o postquirúrgico para ayudar a mantener, o sea, la eubiosis de la boca, además de la utilización, obviamente, en la cavidad del gel, por ejemplo, en el caso de que fuese aprobado, ¿no?, a futuro?
Sí, yo creo que sí, y finalmente hay que hacer una conjunción también con algunas herramientas en las cuales ya se está trabajando, que son también basados en biosensores, de ver, por ejemplo, algunas herramientas que te sensen previo a la colocación del implante, por ejemplo, la presencia, no lo di mucho detalle porque si no me voy a alargar, en relación a que, por ejemplo, virus como Epstein-Barr o citomegalovirus son claramente coinfectantes de nichos de implantes. Y si tú los puedes pesquisar antes de colocar el implante, pudieses hacer incluso una terapia retroviral en eliminar esos virus previo a que tú los coloques el implante, porque es lo que yo decía, no puedes evitar que la saliva se te meta y la sangre y todo, entonces todo va a estar ahí. Entonces, tener esos sensores previos, ¿cierto?, como biomarcadores de potenciales agentes que pudiesen generar un peor pronóstico de tu tratamiento a largo plazo.
Excelente, muchísimas gracias. Y solo recordar que la primera escuela dental en la región fue la de Chile en 1888, impulsada por la Universidad de Chile. Increíble el nivel de investigación que han llegado.
Muchas gracias.
Hay más.
El profesor aquí quería preguntar.
Question? Petar, you have any question? Of course. Well, happy to see you. Yeah.
Thank you very much. It was really interesting for me. Nice presentation. I have few questions about nitrate metabolism. We know that there is a nitrate oxidase, and it forms nitrite, and this nitrite is toxic. So, what do you think are there correlation between oral microbiome that transform nitrate in nitrite and cancer of urinary tracts?
No, I don't hear about that.
Because we know that some diagnostic things that urinary infection, they detect the nitrate, and this indicate that there is infection by some urinary bacteria that transform nitrate to nitrate, nitrate reductase. And I don't know if it could be some connection between this bacteria, but bacteria in mouth, they transform nitrate in nitrite.
I think that is some
Nitrate reductase.
Yes. Escape. Necesito mis lentes. The way is that here. Yeah. NO3.
Nitrate. Yeah. And nitrate oxide. Okay. So this is denitrificated by bacteria.
Yes.
Yeah.
This is a way.
Okay. And other data connecting the HLA system, human histocompatibility system with the diversity of the oral microbiomes.
I think that it's diversity of the
microbiome. Does it depends on HLA, histocompatibility system of the human?
Yeah.
Other some data. I want to.
I have a few that it's a correlation with in a specific of apical periodontitis.
Maybe nobody look at
no,
this.
Yeah. No, I think I try to search something connection.
This
is a good idea.
And what when you talk about chlorhexidine, by chance we in our actinomyces research we try to understand which kind of the carbon sources our actinobacteria can use. We try to use different amino acids, sugars, and so on. And what we found that bacteria can use chlorhexidine for growth.
Yeah, I think that it's a very few amount of actinobacteria in oral space, and then a lot of or the most important actinobacteria is Actinomyces.
Yeah.
That generate infections annual
accelerate
where when put inside of the tooth and going to the root canal to the apis is actinomycosis.
Bovis, Actinomyces bovis is discovered like this. Yeah.
Because this is some surprise to see that is going to this metabolic nitritos, because I think always I think that is a bad bacteria, Actinomyces. I think it depends where you put it. It's going to forward to root canal. Maybe it's a pathogen, but in the oral environment, it's
not dangerous. Yeah. And do you have some whole genome sequence of some bacteria from that we can analyze, see the metabolism?
Yes, I don't show that, but another bacteria found associated to the practice of the dentist in intraoperatory is Cutibacterium acnes. Because where you are work the patients and you touch the skin, touch the files and going to the Cutibacterium is going, and these 4 strains you will, and I do always the chance is very difficult, but we did whole genome sequencing of the all DNA of these strains. They are very, very resistant and generate a possibility of the persistencies in the infections.
So you can cultivate on the
because in the lab we have ¿cómo se dice? Cuando uno tiene las bacterias congeladas. Sí, tenemos, we have a lot of oral strains clinicals, in menos 80, yes.
Okay. Interesting.
Mira, yo tengo una pregunta. Bueno, como todos ya saben, el formar, de alguna forma, yo lo planteo, que el sueño de todo microbio es pegarse en alguna parte. De esta forma, la vida planctónica realmente es una fase de tránsito desde una superficie a otra. En el laboratorio tenemos una distorsión un poco feroz respecto a estudiar un microbio planctónico, cuando en realidad el perfil transcriptómico que presenta cuando está pegado o adherido es diferente. Entonces, cuando hay un experimento o una pregunta que un estudiante le hace a un microbio, lo que viene a continuación, bueno, esa pregunta se la vas a hacer en motivo planctónico o se la vas a hacer en biopelícula. Porque un microbio en biopelícula puede hacer cosas muy distintas. Y puede uno construir un marco teórico hipotético robusto, feliz y muy coherente, pero que en realidad no sucede en la vida real del microbio. Sucede solo en el laboratorio, ¿verdad? Entonces, la formación de biofilm es un problema, bueno, que es ancestral, afecta a muchos aspectos de la vida humana. La clínica tú la has planteado, existen enfermedades, la fibrosis quística, por ejemplo, las infecciones a la piel o respiratorias, todos están asociados a una necesidad que el microbio reconozca una superficie, un receptor, y se pegue, ¿no? ¿Verdad? Y ahí se produce toda la intimidad molecular que cambia. Y eso ocurrió en la industria, por ejemplo, el proceso de fouling y corrosión biogénica también es un problema como tú lo tienes acá. Entonces, se ha investigado, por eso que tú has mencionado a Bill como el promotor inicial, ¿no? De entender este fenómeno de cómo los microbios se pegan. Y dentro de del conocimiento molecular más duro, no es el único, pero el más robusto en datos, es que cuando un microbio sucede que se pega, hay una etapa inicial de reconocimiento inespecífico, en el cual, por ejemplo, una fuerza electrostática puede lograr que el microbio se adhiera. Si el microbio tiene una carga negativa leve en la superficie y tiene una superficie con carga positiva, va a haber una atracción electrostática, pero que es transiente. Cualquier cepillado de dientes lo va a remover de ahí, ¿no? Entonces, cuando ese cepillado de dientes no ocurre, o cuando la limpieza de la cañería no ocurre, viene otra fase en el tiempo que es en el fondo multiplicarse sin despegarse, ¿no? Y comienza a formar una microcolonia, que es la que mostramos. ¿Okay? Y cuando los microbios comienzan a reunirse en poco espacio, comienzan a ocurrir fenómenos de regulación transcripcional basado en densidad celular, que solo ocurren cuando los microbios están en alta densidad, ¿no? Y uno de esos procesos se llama quorum sensing, que en el fondo los microbios sintetizan un metabolito secundario que tiene otro fin dentro de la célula, pero que puede salir. Hay muchos, pero el más famoso fue el primero, que se llama, vamos a decir, lactona, que es una lactona chica, ¿no? Y que entra a otra célula, reconoce una proteína regulatoria y activa todo el repertorio asociado a la síntesis del exopolisacárido.
Claro.
Por eso es que las bacterias planctónicas no fabrican esa molécula, pero cuando se pegan sí lo hacen, y por lo tanto hay una regulación transcripcional dependiente del momento en que se hace, ¿no?
Claro.
Entonces, gran parte de la estrategia de control del biofilm se basó por mucho tiempo, y todavía se hace, con interferir con este mecanismo, que se llama quorum quenching, ¿no? Y el quorum quenching tiene varias formas de interferir. La primera es bloquear la expresión del gen que codifica el autoinductor. La otra es desarmar el autoinductor cuando está en el espacio entre una célula y otra, o sea, una lactonasa, por ejemplo. Y otra es engañar a la proteína regulatoria usando un autoinductor que se parece, pero que hace que la proteína no tenga la vida media necesaria para conversar con el promotor.
Claro.
Entonces, estas técnicas de bloqueo de fouling se basan en este quorum quenching y que hay varios productos comerciales que tienen o una bacteria que produce la enzima o la enzima pura, que son varios tipos, lactonasa, lactasa, etc. O sea, hay como 4 o 5
Presentación.
esquema de interferencia de la señal, ¿no? Entonces la pregunta es, en el fondo, es si en el ámbito clínico tú has explorado o has visto que hay productos de control de biofilm, por ejemplo, usando este tipo de interferencia.
Yo creo que se han intentado, pero no sé si estoy tan clara que tengan claridad que lo hacen, que eso están haciendo. Digamos, lo proponen, pero no...
O sea, una pasta de dientes.
Por ejemplo, yo hoy día mismo en la mañana les hago clase de microbiología a los estudiantes de implantología y encontramos un reporte que usan una glicina. Y esa glicina no se la tiran a la microbiología, se la colocan como un polvillo a la superficie del implante para evitar, entre comillas, este reconocimiento y que no se forme la biopelícula sobre la superficie del implante al momento en que se genera la implantación, digamos, del implante y le colocan estas glicinas. Yo no sé, a lo mejor puede ser, el reporte no dice que está usando ese mecanismo. Y de hecho ellos dicen, como que entienden que son más clínicos y no microbiólogos y dicen, no tenemos claro cómo evita que se cree que, y hablan de que efectivamente ven que hay, al momento que se genera esto y las 3 primeras semanas, hay liberación, por ejemplo, de LPS porque la bacteria se podría estar rompiendo, pero tienen hipótesis, no lo han demostrado del por qué esta glicina, por ejemplo, pegada a la superficie del implante, evita que las bacterias se peguen a la superficie y no generen la biopelícula. Hay estrategia, como le digo, pero tengo claridad de que los colegas, como son clínicos, no saben cómo está funcionando. No los conocen.
Bueno, es común del área de ingeniería también que pasa. Se preocupan por los resultados más que por los mecanismos.
Sí.
Y claro, lo más probable es que esté retardando el proceso, pero no creo que lo vaya a bloquear.
A bloquear, exacto.
¿Hay más preguntas, chicos, sobre la presentación? Si no, le damos las muchas gracias.
Muchísimas
Gracias.
De nada. Ha sido un gusto. He corrido hoy día. Espero que les haya gustado. Si necesitan la presentación, yo se las puedo compartir. El
curso está recién comenzando.
Muchas gracias.
Agradecemos la presentación. Y los dejamos libres de todo pecado hasta las 3. Hoy día, tenemos una conferencia pendiente. Sí. Nos quedó una conferencia pendiente por tiempo que la vamos a reprogramar porque está grabada, así que no va a resistir mucho tiempo. Se va a hacer simple hacer. Pero lo prioritario